Student, today we are going to start dissection of green gram, black gram and soybean. So whatever the purple color flower you are observing here, this is a flower of soybean. Then this is a crop of green gram and this one is a black gram. So common name is green gram, black gram and soybean. See here student, I compiled all three pulse crop, that is soybean, black gram and green gram, in a single table, because whatever the morphology of flower is there and whatever the family is there, then mode of pollination is there, many of the things are same in this crop. So I made a comparative table here for your easy understanding, and I merged these three crops in a single video. Because of whatever the dissection of these three flowers are there, these three flowers dissection are same, because whole of the morphology of these three flowers are same. So I compiled, so I shuffled these three crops in a single video. So botanical name of green gram is Vigna radiata, then black gram Vigna mungo, and the soybean glycine max. Chromosome number 2n is equal to 22. And black gram chromosome number 2n is equal to 22 and soybean chromosome number 2n is equal to 40. The three crops belongs to Leguminaceae family, and the subfamily I have mentioned here subfamily because this is important for your exam point of view. Sometimes the examiners ask fill in the blanks or sub question for spotting that mention. Write down subfamily of Leguminaceae or subfamily of green gram, black gram, soybean, pigeon pea, any of the pulse crop which belongs to the Leguminaceae family. So the subfamily is the Papilionaceae, and the mode of pollination in these three crops are the cell pollination. Why it is cell pollination? Because whatever the Androecium and gynoeciums are enclosed in a structure which is called as a keel, and the cleistogamy condition which favors the cell pollination is in these three crops. Then see here the flower type. The flower of these three crops are the perfect one. What is meant by perfect flower? The flower which contains Androecium and gynoecium is called as perfect flower. Then the flower is complete one. Why it is complete one? Because it contains calyx, corolla, Androecium and the gynoecium. Those flower which possess Androecium, gynoecium, calyx, corolla, then this flower is called as a complete flower. Then estivation means it is the arrangement of the petal. Okay, earlier class I have mentioned that the estivation, that is twisted estivation I have mentioned, velvet type of estivation I have mentioned there. It is the arrangement of the petal in which the flowers, the petals of the flowers will not overlap each other. They join by their edges is called as a velvet type of estivation. And if the petals of the flowers, they overlap each other, as like which is present in bhendi flower or in cotton flower, then this type of the estivation is called as a twisted estivation. And the third one type of estivation present in the flower crop, that is a vexillary estivation. Vexillary estivation means what? This vexillary estivation, there is a arrangement of 5 petals are there. The largest one which is outside of that flower is called as a standard petal. If you see here, this one is a standard. Then inside the standard petal there is a 2 wings are present. It is separated from here. The 2 wings are present here, and inside the wing there is a 2 keels are present there. Okay, 2 keel petals are present there. Then this type of the estivation present in pulse crop is called as vexillary estivation, and it is also important for your objective point of view. So the definition of maxillary estivation is that the posterior largest petal, that is the standard, whatever the largest petal is there, which is outside, which is outermost petal is there, it is called as a standard petal, which overlap two lateral petals. It overlaps two lateral petals, it is called as wing petal, and which in turns overlap to anterior petals, it is called as a keel. So the arrangement of the pulse family flower is as like that. There is one standard, two wings and one keel. Standard encloses two wings and wings encloses two keels. This arrangement of the petals is called as maxillary estivation. Okay, so in tomato brinjal we see the velvet type of estivation. In okra and in cotton crop there will be a twisted estivation, and in pulse family crop there will be a maxillary estivation. Then calyx, the condition of calyx. Okay, and this is the morphology of pulse family flowers, which is same in all the crops, except there is a only difference in case of groundnut, that is in case of Androecium. Okay, so whatever, when you take any pulse crop, then the morphology of petals is same. First one is standard, wings and keels, but inside the keels, whatever the Androeciums and gynoeciums are enclosed there, and these the 10 number of anthers present in the pulse family, and the condition of anthers is diadalpous. What is meant by diadalpous? There is a bunch of 10 stamens are there, and one free anther, one free stamens arising from that bunch of 9 anthers are there. Okay, so this condition is called as diadalpous condition, and most of the pulse family crop possess diadalpous condition of anther except groundnut, because in groundnut monoadalpous condition is there, and groundnut there is a 10 bunch of anthers is there. Okay, whatever the one anther is there, that one is a sterile one, and there is a 9 bunch of anther which encloses inside the gynoecium. Okay, so see here, this is the structure. The gynoecium is enclosed inside the 9 anthers, and one stamen is emerged out from that bunch of 10 anthers. So this condition is called as a this condition is called as a diadalpous condition of Androecium. Okay, now we covered this point. Diadalpous condition, 10 stamens are there. What is meant by diadalpous condition? The 10 stamens divided into 9 stamens bunch, and one free stamen is there from the base of that 9 stamens, the bunch of 9 stamens. So this condition is called as a diadalpous condition. And then the corolla condition. Corolla means petals, so the arrangement of the corolla is one standard, two wings and two keels, and inside the keel there is Androecium and gynoecium. The calyx, the condition of calyx is also, sorry, I have made this should not be there. The condition of the calyx is gamosepalous condition. So because the calyx is fused together, so the condition of calyx is gamosepalous. And the gynoecium in case of green gram and black gram, the gynoecium in case of green gram, black gram, and in case of soybean, it is a superior. What is meant by superior? The ovary, the arrangement of ovary is to the top, and whatever the another walls that are arising from the base of that ovary, then this condition of ovary is called as a superior ovary, or it is also called as a hypogynous ovary. Okay, then in case of green gram and black gram, the condition of gynoecium is a monocapillary. What is meant by monocapillary? When we cannot differentiate into ovule, then style and the stigma, then this condition is called as a monocapillary condition. It is a slender one, and we cannot differentiate into a ovule, style and the stigma. So the gynoecium present, this is the flower of green gram. So the whatever the gynoecium, whatever the ovary does not be specified or differentiated into different parts. So the gynoecium is a monocapillary in case of green gram and black gram, while in case of soybean, the condition of gynoecium is a capitate. Capitate means what? Capitate means what? It possesses a head-like structure. The stigma possesses a head-like structure there, then it is called as a capitate stigma. So the gynoecium in case of green gram, black gram is superior and monocapillary, and in case of soybean, superior and capitate gynoecium present there. Okay, then next one. This is the floral biology of soybean and green gram. Here you can easily identify the different parts. The outermost big petal is called as standard. Then inside the standard there is a two wings are present. Inside the wing there will be there are two keels are present, and inside the keels there is this structure is present. This structure where the one stamen is arising from the base of 9 stamens. So this condition is called as diadelphous condition, and this is the calyx. You can easily observe here the calyx is fused together, so the condition of calyx is a gamosepalous condition. Okay, and this is the flower of mung bean. Here the outermost is standard, then here are two wings are present, and the innermost here is two keels are present, and inside the keels the androeciums, and inside the androecium there is a gynoecium. The condition of androecium here is diadelphous, the one stamens arising from the base of 9 anthers. Okay, so this is the floral biology. This is the floral biology of soybean and green gram. Exactly the same floral biology of black gram flower is there, so I have not mentioned here. Then the next point is that emasculation and pollination in green gram. While going for the hybridization for create a variability, firstly we have to select a bird which will be open on a tomorrow. So this condition of bird is called as a hood stage of the bird. So select this condition of the bird, then open, then remove the calyx and open the bird, open the petals of the bird, and then remove that petals with due care without injuring a gynoecium. Okay, then after removing these petals there will be a structure. There is a bunch of 10 anthers, and inside that 10 anthers there is a gynoecium. The care should be taken that no single anther should be bursted here. No single anther should not be form pollen powder there. Okay, if the condition is that, then it may self-pollinate the gynoecium. So you should select a bird in which the anthers will not be a matured one. Okay, then remove these anthers with the help of forceps or the needle. Then whatever the remaining structure is there, that one is the gynoecium. Okay, the gynoecium only remain here. After gynoecium, the gynoecium is so small in this green gram, black gram and soybean, so there is no need to bag it with the butter paper bag. Only for the identification you put a thread there, you put a tag there, and write down the information of male parent and female parent there. Okay, after that the most important step is that the pollination. So whatever the pollen grains, whatever the male parents is there, you take a flower from there, open flower from there, where the pollen grains are formed, where the pollen grains are viable, and then remove the whatever the standard wings and keels there. Only take out that the bunch of 10 anthers, and then rub this powder on that gynoecium. So this is the pollination. Okay, so these are the different steps which are used for the emasculation and pollination of green gram. Then this one is the emasculation of, now we see the emasculation, how to do a emasculation and pollination in case of soybean. It is exactly same as that of the green gram. Firstly you have to select a twig, you have to select a peduncle or a branch where you have to you are interested to do a emasculation or the pollination. And while you are going for emasculation, at that time you should pick out or the remove whatever the adult flowers are there, and so immature birds are there. Keep only 2 to 3 hood stage birds on which you are interested to do a emasculation there. After that, here this condition is that here the adult floret are removed by that person, and here the proper stage hood condition of bird is selected here. After that, here there is a opening of that bud, opening of bud should be done there, and after opening of that bud you should remove that standard wings and keels from that bud. Then the whatever the remaining structure is there, that one that will be a here the whatever the standard wings and keels removed here, and also whatever the anthers are there, that anthers are removed here, and the remaining structure, see here the gynoecium. Pubescence is also present on a soybean plant, and the remaining structure is that only the gynoecium. So the emasculation is done here. What is meant by emasculation? It is the removal of anthers, or it is removal of stamens without injuring gynoecium, is called as a emasculation. Now the emasculation is done in the soybean. What is the steps? Firstly, you have to remove the adult or the mature floret or the mature flower from that branch. Then take up a hood stage condition of a bud. Then open it with the help of forceps or the needle. Then remove the whatever the petals are there. Then tag it with a well labeled. After tagging, you have to remove all the petals, all the 9+1 anthers from that bud, and whatever the remaining structure is there, that is the gynoecium. Okay. Then after that, the pollination in soybean. How to do a pollination? For pollinating, as the soybean flower is so small, so select a mature flower there and remove it from the plant. After inside the keel there is a Androecium is present. So open the flower with the help of forceps, then remove the whatever the petals are there, and take out that 10 anthers with the help of forceps. Remove that 10 anthers and gently rub these Androeciums on emasculated gynoecium. Okay. After that, you have to take a proper thread or the tag on that emasculated flower, because after seed set we have to identify on which flower we done hybridization. For that identification purpose, the tagging should be done there. After tagging, at the mature stage it will be looks like, and this will be a crossed pod there. So this is the pollination in case of soybean. In that way, this is the emasculation and pollination in case of green gram, and same will be followed in case of black gram, and same procedure is repeated for soybean. So this is the procedure, stepwise procedure of emasculation, and this is stepwise procedure of pollination in case of soybean. Okay. So for soybean, these are the dissected part. First one is a bract which is present on that calyx, then this one is a keels, then these are the wings, and this one is a standard. This is 9+1 condition, and this is a gynoecium. And this is the mounted parts of soybean, and this is the diagram which I have shared in your practical manual PDF also. So you take a reference from there and you should mount these parts as like that. Then this is the dissection of green gram and the black gram. The dissection is exactly same in both the crops, because both the crops possesses same floral biology. First one is a calyx, then keels, wings, standard, and this one is a 9+1 condition. One anther is here. You should keep a distance between 9+1 anthers. Okay. You should keep some space while sticking these parts as like that. Okay. This one is 9 and this one is 1. So Androecium, the condition of Androecium is diadalpous, and this one is a gynoecium, which will be gynoecium is monocapillary and sessile. And monocapillary means we cannot differentiate this gynoecium into a ovary, style and the stigma. So the gynoecium is a capillary one. In that way you should mount the different parts of your green gram and black gram. Then this is the assignment for you. Do a dissection of green gram, black gram, soybean flower and mount different parts in a sequence on mounting sheet. After that you have to draw a neat label diagram of floral biology of soybean crop. Okay, so this is the assignment for you on this practical exercise, and see upcoming video carefully and do a practice of dissection of flower. Good morning. Today we are going to start dissection of urad bean and mung bean. Okay. As we know that the flower morphology and the flower structure and the appearance of the urad and mung are similar. Only there is a difference in taste and whatever the stickiness in the in case of the urad grain are more sticky after cooking. But this stickiness is not observed in case of green gram. So the both the pulses having a pod shape same, and grain size, grain morphology also same in these two pulses. Only the difference in case of flower is that the green gram flower's color is green color. It is dull green in color, while the flower color of that urad bean is bright yellow color. So on the basis of flower you can identify the plant also. So the color of green gram is dull greenish color. See here, the dull yellow or the dull grayish color is present here, and the color of urad bean is bright yellow in color. So this is the main difference in case of the flower color. But whole other morphology or the different parts of the flower is the same. As I said that in case of pulses there is a vexillary activation is present. In case of pulses, vexillary activation is present. In pulses 5 petals are present there, and these 5 petals are arranged as like that. The outermost is standard petal. After that there is a pair of keels, there is pair of wings. Now after wings, the innermost portion where in which the Androecium and gynoecium are enclosed, this condition is called as the pair of petals is called as a keels. The spelling of keel is K-E-E-L-S, not K-I-L-L-S. Okay. Then there is a two condition in case of pulses. If the 10 number of anthers present together, only 10 number of anthers present into the pulses, and if that 10 number of anthers forming a bunch, then it is called as a monoidalpous condition, and which is found in the groundnut only. Okay. And if that 10 anthers splits into 9+1 condition, that 9+1 condition means the 9 anthers forming a single bunch, and the remaining one forming the remaining one anther arising from that bunch is called as a 9+1 condition, and it is called as a diadalpous condition. And this diadalpous condition is found in chickpea, pigeon pea, then urad, mung. Only the example of monoidalpous is the groundnut. Okay, so the rest of the pulses showing 9+1 condition. If you observe here, so if you observe here, I have separated a bunch of 9+1 anthers here. So from the base, the one anther is arising from the bunch of 9 anthers. If you count these anthers, then it will be a only 9s, and one is arising from the base. So the care should be taken while removing keels, that the one anther remains with the bunch of that 9 anthers. Okay, so this is the 9+1 condition. If I have separated this one is the one number of anther, and the remaining one, these are the 9 number of the anthers. While removing the gynoecium from that, from that 9 plus 1 anthers, in 9 bunch, in 9 number of anthers, there is a presence of gynoecium, and which is one which is somewhat feathery, and you can identify because it is somewhat thick than that of the anthers. Okay, see here, this is the gynoecium. So while removing gynoecium, give a gentle cut from the back of that bunch of anthers and separate out it up to the top. Okay, now I have removed, and then take out this greenish color gynoecium from the bunch of 9 anthers. Now I am taking out it. Okay, now my gynoecium is separated. This is gynoecium. Okay, and this is the 9 anthers. So you should understand the condition in case of urad bean. This is the flower which I have dissected here. This is the flower of urad bean, and the condition of anthers is 9 plus 1, so it is the diadalpous condition, and this is the gynoecium. Okay, this is the gynoecium, which is the appearance of gynoecium is somewhat thick, so it is easy to identify gynoecium in the bunch of 9 anthers. The one anther which is arising from the base of 9 anthers is a sterile one, so it don't have any function in case of pollination. Okay, now we will start a dissection of flower. I will show you only one dissection. If possible, I will cover a green gram, because both the flowers dissection is the same. If I do a dissection of urad bean, the same dissection will be followed for the green gram. Okay, so let's start our dissection of urad bean. Only you should understand that the presence of 9 plus 1 condition in case of urad bean and the green gram. So the one anther is separated from the bunch of 9 anthers, and inside that this gynoecium is present. Now we start our dissection. How to dissect a flower? Firstly, you have to remove this calyx. The condition of the calyx is both the calyx are fused together, so the condition is gamosepalous condition in case of urad bean and the green gram, and the pubescence is also, or the hairs like structure also present on the calyx, so the calyx is gamosepalous in case of urad bean. So now we start a dissection of urad bean. Firstly, you have to remove calyx. Okay, the flower is somewhat tough, so give a gentle cut. Give a gentle cut to calyx. Okay. Then with the help of forceps, remove it from both the side. The gynoecium is attached to the calyx, so the care should be taken that it should be free from the base of gynoecium and the bunch of anthers. If it get attached, then our 9 plus 1 anther get missed there. So the calyx, I have removed the calyx here. Okay, then you have to separate out standard petal. Standard. You can paste your part in either side of that plant part or the petals. You can follow this, or you can invert this. After that, after wing there is a presence of pair of after standard petal there is a presence of wings. So I have removed one wing here. I will place the same here. Okay, then remove the other side of the wing. Now the other side of the wing is removed by me. Then see here, the keels are present here and keels are free from each other. If you see here, the keels are free from each other. Then remove it, and either you can give a cut to the these keels first and then remove from the base of that flower, or otherwise remove the cone shape of that keels and then give a cut to keels. Okay, then the keels are separated from each other. Okay, the keels are separated from each other, and I have to arrange the same here. Okay, the 9+1 condition, where one okay, our one number of anther is here. Okay, where it goes? Okay, it falls down. I have taken it from the base. The one number of anther is there and it is so much thin, so sometimes it get attached to the keels, or sometimes it get attached to the calyx. So you should take care that it should not be removed removed with any other part like standard wings or the keels. It should remain with the 9+9 bunch of the anthers. Now we have to remove gynoecium. Gynoecium, which is green in color and which is thick in appearance, so it is easy in case of wooded bean to identify our gynoecium. Give a cut from the back side to that 9 bunch of the anthers and then separate out our gynoecium. Okay, now it is separated, and the care should be taken that the gynoecium should not be break into a pieces. Okay, now how to arrange this dissection? Firstly, arrange your gynoecium because it is the innermost part. This is the gynoecium of wooded bean. Then the 9+1 condition of the anthers. You should keep a space between these two parts. 9 plus, it should be identified into a pasted dissection. So you should keep some distance between these two, the one anther separated and 9 anthers separated. After that, standard will be there. After standard, you should arrange wings. After wings, arrange keels. Okay, the direction should be same and the tip should be on upside and the point of attachment should be downside. Okay, then these are the okay, now it is arranged. The part of wooded bean and green gram is somewhat not plain, so it is hard to paste this part on mounting sheet. So this is all about the dissection of your green gram, and the same dissection is followed for sorry, this is dissection of urad bean, and the same dissection will be there for green gram. So I am showing you once the green gram dissection here, the 9+1 condition, then gynoecium, and the same will be followed for your practical. Thank you. Now we start the dissection of green gram flower as like same the as like in case of urad bean. Give a vertical cut, then give a round cut to your calyx and remove same from the base of that flower in fused condition. Okay, this is the calyx of your green gram. This is calyx, then remove standard petal. This is standard, then remove wings. Oh, so nice. The creativity of God is so nice. The mode of pollination is also depends upon that. See here, because the Androecium and gynoecium are enclosed into the keels, so the foreign pollen grains can't reach up to the gynoecium, and which facilitates cell pollination in case of urad bean, then green gram, then groundnut. See here the very well 9+1 condition. The one anther it is attached at the base of that, and this is the bunch of 9 anthers. And now separate out our gynoecium. Same, the cut should be given from the back of that 9 bunch of the anther. And then gently pick up, and the care should be taken that it should not be cut into the pieces. And by mistake I have cut my gynoecium here. So as like in the urad bean, you should follow the same guidelines. It should not be cut into the pieces. The gynoecium should be from the ovule style and the stigma. If in any case it get break, then use another one flower. The gynoecium will be as like. Okay, it should not be cut into the pieces. So this is the dissection of green gram. The arrangement is same as that of the urad bean. Thank you. Good morning, student. Today we are going to start dissection of soybean flower. The color of flower is purple color, and the condition of calyx is fused together. So the condition of calyx is called as a gamosepalous condition. On calyx there is a presence of 2 bracts, and the 5 petals are present together. The one petal is called the outermost petal is called as a standard petal. Inside the standard petal there is a presence of 2 wings petals are present, and inside the wing 2 keels are present. In that keels the Androecium and gynoecium are enclosed, and the keels avoid the entry of foreign pollen grain on gynoecium. So the mode of pollination is cell pollination. It strictly avoid the cross pollination. The 10 number of anthers present in soybean, and 9 number of anthers forming a bunch, and one separate anther is arising from the base of that bunch is called this condition. The 9+1 condition is called as a diadalpous condition, and inside this bunch the gynoecium is present. Such diadelphous condition is also found in the pulses like green gram, urad bean, pigeon pea and the soybean. If the 9 plus 1, if the 10 anthers present together in a single bunch, then this condition is called as a monoadelphous condition, and this monoadelphous condition found in the groundnut crop. So where that bracts are present, if you observe here in this flower, the bracts are present on this calyx. I am showing it with the help of needle. On side by two, on opposite side of the calyx, the two bracts are present. Okay, these are the bracts which are present on that calyx. So the condition of calyx, here the calyx is fused together. So the condition of calyx is gamocephalous, and on calyx two bracts are present. So now we start the dissection of soybean flower. Here firstly you have to remove the bract first. The two bracts present on calyx opposite to each other. Then the flower is too delicate, so do dissection very carefully and gently. Give a vertical cut to a calyx, then slightly twist a flower. And made a base free. Now standard is removed here. Now our calyx is also removed here. The care should be taken that the 9 plus 1 condition of anther, sometimes the anther get attached to anywhere to the calyx. Okay, then remove. Very nice. Then these are this one is wings, and the innermost two petals are the keels. Now these are the wings. Arrange it in the inward position. And these one are the keels. The keels are fused together at the top, so with the help of needle you can make it separate. Then separate it with the help of needle and arrange it in a inward direction. Okay. See here the 9 plus 1 condition of the anther I am showing to you. See carefully, the thread like structure is present here, and one single anther is present at the base. See here, you can see here very carefully that one anther is attached to the base. I am going to separate out this anther. The anther is so delicate and tiny, so remove it with the help with two care. Okay. Now it get removed. See here, this one is a 9+1 condition of the anther. Now place this 9+1 condition of the anther. One anther here. As like in the mung, urad, same dissection is in case of soybean, but only the thing is that the parts are too delicate and too small, so it is somewhat hard to handle the flower. Then remove gynoecium. This one is 9 anthers bunch, and then remove gynoecium. Okay. Now this is the dissection of soybean flower. It is so easy. Simply you have to remove the standard. Okay. Then the wings, and then keels, and the innermost portion which is remaining here is your Androecium and gynoecium, and here at a base and one single anther is get attached. See here, one anther gets separated, this one. So, and inside the bunch of 9 anther there is a presence of gynoecium, which is greenish one. Then remove this gynoecium. So easy dissection is this. So in that way you proceed for your dissection and paste a different part of soybean on a mounting sheet. Thank you.